Mitosis in somatic cells is eventually going to lead to cytokinesis and the development of two hopefully identical daughter cells. This is a form of asexual reproduction. Same if you're a prokaryote and you undergo binary fission to create two daughter cells, another form of asexual reproduction. So there is no fusing of nuclei together or sharing of genetic material in order to create a new organism. You are just simply dividing and hopefully creating an exact copy of the parent cell. Any variations that happen is going to typically be something exogenous or due to something exogenous or outside of the cell. Things like mutations or environmental effects can affect the variation in the daughter cells, but it is not a self-imposed mechanism. So when looking at the human genome, we are going to have 22 pairs of somatic chromosomes. So somatic chromosomes, that's going to be your chromosome number 1 through your chromosome number 22, meaning you have 44 total. If you have 22 pairs of something, 2 of each, that means you have 44 in total. You also have 1 pair of sex chromosomes, so therefore you have 2 total sex chromosomes in your somatic cells. Now, altogether, so we're going to combine the sex chromosomes as well as the somatic chromosomes. Altogether, you're going to have 23 pairs of chromosomes in all of your somatic cells, therefore giving all of your diploid somatic cells 46 total chromosomes. So here, you have paternal chromosome number 1 and maternal chromosome number 1. For your chromosome number 2, paternal chromosome number 2, maternal chromosome number 2. For chromosome number 3, paternal chromosome number 3, maternal chromosome number 3. So each of these are pairs of somatic chromosomes. Pairs of somatic chromosomes, meaning, let's say this was a hypothetical alien cell containing 3 pairs of chromosomes, somatic chromosomes. Well, therefore, it would have a total of 6 chromosomes. All right. So we have 23 homologous pairs in total, meaning we're diploid. Or actually, our somatic cells are all diploid. So what diploid means is that you have 2 chromosomes of each chromosome number for every chromosome number. So both chromosomes of a pair. So for example, your chromosome number 21, you're going to have 1 maternal chromosome number 21 and 1 paternal chromosome number 21 in your somatic cells. Now when we're talking about haploid cells, which are going to be our sex cells, they're going to have only 1 chromosome of a pair. So for example, only 1 chromosome number 21, and that could either be maternal or it could be paternal. All right, so mitosis, the goal of mitosis is to go from a diploid cell, to another diploid cell, or to 2 diploid cells. You're going from 2N to 2N. However, when we're dealing with meiosis, we're going from a diploid cell to a haploid cell. So we're going from a 2N to 1N in meiosis. During mitosis, you're going from 2N to 2N. All right, so karyotypes, that is just a snapshot of all of your chromosomes during metaphase of mitosis. So a photo of a cell frozen in metaphase. And what we do is we actually order our chromosomes based on size. So our largest chromosome, chromosome number 1, well, it's number 1 because it's the largest chromosome. And we do this for all of the somatic chromosomes. So chromosome number 1 is going to be the largest, number 2 is going to be the second largest, number 3 is going to be the third largest, all the way till we get to chromosome number 22, because within our somatic chromosomes, both our paternal and our maternal are going to be the same size. However, when we get to our sex chromosomes, the Y chromosome is actually a lot smaller than the X chromosome. In this case, you have 2 X chromosomes, making this organism biological female, right? So you have 2 X chromosomes, they're going to be the same size. However, if it was a male, you would have 1 X chromosome and 1 Y chromosome, the Y chromosome being much smaller. All right, so now let's talk about sexual reproduction. Meiosis is the formation of, you could say, gamete nuclei, and then eventually cytokinesis creates those new individual cells. So meiosis is is a main player in gamete production. Your gametes are either going to be your sperm or your eggs. And typically, when you create a new organism, a zygote, two parents are going to contribute their own set of chromosomes. Remember, they're going to be haploid. So you're going to have one gamete, one haploid gamete, an egg, plus one haploid gamete, a sperm, making one diploid zygote. So the purpose of meiosis, you have a lot of self-imposed mechanisms to create variation. So the purpose of meiosis is to create variety that are beyond mutations or just the environment. So meiosis is going to consist of two nuclear divisions rather than the one that you would see in mitosis. So DNA is only going to be replicated once in S phase. So if you remember, during interphase, you undergo this process or this phase called S phase, and that's where you synthesize new DNA. So that's the only time that DNA is going to be replicated. However, you're going to have two nuclear divisions, meaning all of your daughter cells are going to be haploid, or 1N. So the functions of meiosis is to go from 2N, from a diploid cell to a haploid cell, and again, you're creating genetic diversity. So to jump right into it, we're going to start off with prophase 1. So a lot like prophase of mitosis, what's going to happen is your nuclear envelope is going to dissolve and your chromosomes are going to condense. Now what makes this a little bit different from mitosis, or prophase of mitosis, is that during prophase 1 of meiosis, you get this process called synapsis. And so synapsis is the movement or the coming together of all of your homologous pairs. Remember, we are starting off with a diploid cell, meaning you have two of every chromosome number. So what happens during synapsis is all of your chromosomes of the same chromosome number move together to become within proximity of one another. So your homologous pairs come together. So in this case, you have your chromosome, your maternal chromosome number 1, your paternal chromosome number 1, your maternal chromosome number 2, pairing up with your paternal chromosome number 2. So this would form tetrads. Tetra meaning 4, and if you look at these chromosomes, they're bilobed. So you have one lobe, two lobes in this chromosome, one lobe, two lobes in this chromosome, and when two of those bilobed chromosomes come together, you get 1, 2, 3, 4 lobes. So you form tetrads. So again, your nuclear envelope dissolves, chromosomes condense, and the first thing that happens is this process called synapsis, which brings your homologous pairs together to form a tetrad. Now, once those tetrads are formed, what you're going to get is a process called crossing over. So this is the first process that's actually going to be that self-induced creation of genetic variation. We call this crossing over. So crossing over is where once you form those tetrads, you actually swap genetic material between those homologous pairs. So in this case, you have your maternal chromosome number 1 and then your paternal chromosome number 1 here. They form this tetrad, and what's going to happen is crossing over occurs, and now you have this modified version of chromosome number 1 from your mom and this modified chromosome number 1 from your dad. What we would call those modifications are recombinant chromatids. So remember, this chromosome is composed of two sister chromatids, one of them being a recombinant chromatid because it has modified itself by swapping genetic material with another chromatid. So here we have, or during prophase 1, nuclear envelope dissolves, chromosomes condense, synapsis occurs, forming tetrads for every chromosome number. Also keep in mind, we're looking at a cell that is only depicting two chromosome numbers, right? You have chromosome number 1, chromosome number 2, but in our case, within our cells, we're doing this for 23 homologous pairs. So this is happening for all of the chromosomes simultaneously. This is just a simplified version. So here you have crossing over that's occurred, and now you have these recombinant or modified chromatids. So still prophase 1. Now moving to metaphase 1, this is another during metaphase one, you're going to have another mechanism that's going to contribute to genetic diversity. So what happens is your homologous pairs are going to line up down the middle. So if you remember in mitosis, what happens is during metaphase of mitosis, all of your chromosomes line up single file down the middle. However, because we're dealing with meiosis, during metaphase 1 of meiosis, things are going to line up down the middle. It's just different things are going to line up down the middle. Instead of all of our chromosomes lining up single file, what happens is our chromosomes line up as homologous pairs. And so our homologous pairs will line up down the middle, or those tetrads will line up down the middle. And what happens is you get this thing called independent assortment. And so if you look at this first cell here, and again, we're dealing with many, many, many chromosomes, not just 2 chromosome numbers. So we're dealing with more pairs than just 2, but this is just a simplified version. You have your dad's chromosome number 1 and your dad's chromosome number 2, both moving to the left side. So both of your dad's chromosomes, chromosome number 1 and chromosome number 2, move to the left side, whereas your mom's chromosome number 1 and chromosome number 2 move to the right side. However, equally and justifiably, you could have, those chromosomes, those same chromosomes moving to a different location within the cell. So in this case, you could have, your paternal chromosome number 1 grouping up with your maternal chromosome number 2, and so on. You have to think about this in terms of every other chromosome that's happening within our cells. And so as a result, what would happen is this simple independent assortment of these individual chromosomes are going to lead to different combinations of, you could say, chromosome distribution. Now, to add more layers to this, some of your homologous pairs during prophase 1 will create recombinants, or some of your chromosomes will create or will cross over, some will not. And those variations are going to, in a sense, be compiled with the swapping of these chromosomes. And you could even think of it as though the genes themselves, or you could say the alleles themselves, are independently assorting. So again, this is kind of a simplified version. This is not taking into account, the variation that you could get from the crossing over that would eventually lead to even more variation than just these simple, clean examples of independent assortment. Each gene, or you could say each allele, is independently assorting through that process. So just to recap, the two main contributors to genetic diversity are going to happen within the first 2 phases of meiosis. During prophase 1, you get crossing over, and then still during prophase 1 of meiosis 1, you're going to get crossing over, and then during metaphase 1 of meiosis 1, you're going to get independent assortment. So after your metaphase 1 of meiosis, you have anaphase 1. So I like to associate the A with a part. Depending on what you're talking about, whether it's mitosis or meiosis 1 or meiosis 2, something's going to be moving apart. It's just a matter of what is moving apart. So during anaphase 1 of meiosis, each of your chromosomes, and I put this little X here to depict that they are bilobed. So each of your bilobed X-shaped chromosomes, X-shaped condensed chromosomes, are going to separate from their homologue. So each chromosome is going to separate from their homologue. The homologous pairs are what are going to be separating. You could say the tetrads are going to be separating. So the potentially modified paternal chromosome number 1 will separate from the potentially modified maternal chromosome number 1, and so on for every chromosome number. So after anaphase, you will undergo telophase, and so during telophase, each chromosome is, well, it's technically going to be different, or most of the chromosomes are going to be different from their parent chromosomes. All right. So during meiosis 2, this is going to be the second division. And actually, I forgot to mention. So during, right after telophase 1, you're going to get a cytokinesis. You get that cleavage furrow, and you'll eventually get a cytokinesis. And now we're dealing with 2 individual cells. So now during meiosis 2, we're going to be dealing with the second division. Remember, meiosis has two nuclear and cellular divisions, whereas mitosis only has one nuclear and cellular division. All right, so meiosis 2 is going to be a lot like mitosis, but only one of each chromosome number per cell at the very end. So kind of like in mitosis, prophase of mitosis, your nuclear envelope dissolves, it's going to be non-existent, and your chromosomes will be condensed. So prophase 2, a lot like prophase of mitosis, you don't have any weird processes that happen to, increase genetic variation. It is pretty straightforward step. Then you move on to metaphase 2. So during metaphase 2, remember we're dealing with two cells, and if you look here, this is your modified chromosome number 1 from your mom. Its homologue is actually on a completely different cell. And so what's happening during metaphase 2 is all of the chromosomes that you have are going to line up down the middle. They're not lining up down the middle as tetrads. They're actually, it's going to more closely resemble mitosis in that all of the chromosomes you have present in each of these individual cells are going to line up single file down the middle of the cell. So all of your chromosomes line up down the middle, including the recombinant chromosomes. And then during anaphase 2 of meiosis 2, what happens is your sister chromatids will separate, a lot like during mitosis. So your sister chromatids will separate and move to each pole of these individual cells. And then you have telophase 2, where you get the reformation of your nuclear envelope and then the decondensing of your chromosomes back into chromatin form. And so at the very end of all of these subphases, you end up with 4 haploid cells, or 1N cells, meaning they have one of every chromosome number. All right, so meiosis is going to lead to genetic diversity. One of the players is going to be crossing over after synapsis during prophase 1, and then the other player is going to be independent assortment during metaphase 1. And this is just where you have the equal chance of chromatids to move to either of the 4 different gametes at the very end of it. All right, so let's talk about some meiotic errors that can occur. Non-disjunction, kind of as the name implies, it's non, so the absence of disjoining, so things failing to disjoin, so that this is where you have no separation in an anaphase step. So more common in anaphase 2. However, this picture here, this figure, actually depicts this happening during anaphase 1. So we'll just go over it really quick. So you have meiosis 1. During meiosis 1, remember your homologous pairs are supposed to separate during anaphase 1 of meiosis 1. But in this case, let's say they don't. They just remain as a tetrad, or they remain together, and they move, both of them move simultaneously to one side of the cell, leaving the other cell deficient in one of those chromosomes altogether. And that's depicted by this dotted line. So this is actually missing. We'll just say that this cell here is missing chromosome number 21 altogether. And then this cell actually has 2 chromosome number 21s, whereas it's supposed to only have 1 on this side, but instead it has 2. Now, if we take it further, so we're going to complete meiosis. What happens is this cell completes meiosis, creating these cells that are supposed to have one of every chromosome number. So they're supposed to have 1 chromosome number 1 from either mom or dad, 1 chromosome number 2 from either mom or dad, and so on, up until you get to, chromosome number 21, and you, instead of having 1 chromosome number 21, you have 2. Now when you have that egg, we'll just say this is an egg, for example, and it fuses with a sperm that contains its own chromosome number 21, well, now you've created a zygote with 3 chromosome number 21s. Whereas this cell over here, after the formation of this zygote, it only has 1 chromosome number 21. So we would actually call this this mechanism, or you could call this a description of this would be aneuploidy. So this is just where you get a different chromosome number than your parent. So a prime example of this, a really common example of this, is actually trisomy 21. Tri meaning 3, trisomy 21. So if you look at the karyotype here, this organism is going to have 3 chromosome number 21, meaning it has, well, it has 3 chromosome number 21 rather than 2, which is different from its parent, therefore making it a form of aneuploidy. Now, polyploidy is where you actually get an extra set of every chromosome number. So, for example, let's say you had 3 of every chromosome. That would mean you were a polyploid, which is actually very common in plants, or at least more common in plants than it is in animals. Okay. So last, let's talk about different ways your cell can die, or cell death. So cells can die typically in 2 ways. So the first one is necrosis. So this is kind of like a natural causes of death. You could have poison, or your cell itself could starve to death. We would call that necrosis. So typically with necrosis, you get cell swelling, and the plasma membrane will rupture, releasing all of its compounds and components into whatever tissue it ruptures within. Oftentimes, this is going to cause or lead to inflammation. All of the cytokines and cell signaling molecules within that cell are just released, and it kind of wreaks some havoc within that tissue. However, another type of death is programmed cell death. This is where a cell would be potentially programmed to die, whether it's not needed, it's old, it's dysfunctional, or even if it's helping in in forming the morphology of the multicellular organism. Some tissue needs to die off in order to create certain morphologies. And so this is just, programmed cell death. And so what happens is you actually get what we call membrane blebbing. This is actually where the cytoskeleton will dissociate from the cell membrane, creating like lumps and crevices within the cell. And eventually the cell will break apart into apoptotic bodies. So it creates like these little compartments of their organelles and pieces that can easily be phagocytosed or eaten by neighboring cells. Typically, this is not going to lead to inflammation, because you're not just rupturing your cell membrane, releasing all of its contents into the tissue. What you're doing is you're creating these nice little packages in vesicles that can be consumed by other cells.